My background is in the area of aquatic ecology. I'm mostly dabble and play in ecosystems. I'm an ecologist, but I'm also an ecotoxicologist. So I study the science of poisons, and one of the poisons that I've been studying most recently, the last 5 or 7 years or so, has been toxin-producing algae, and in particular the toxins. The talk that I'm going to talk about or introduce you to today, the subject, the harmful algal blooms, I like to think that I operate under the lens of One Health. I've often thought about the work that I do in that context, but like most of us, we don't infuse or incorporate every part of the tripartite relationship, animal, human, environmental health in what we do. But through our interactions with others, we often do touch upon all of those. And I will talk about all components in today's talk, and in particular on the ecosystem and human health side, because that's the two areas that I've spent most of my time in with respect to harmful algal blooms. So by way of a presentation overview, I'll give a very brief history of algal blooms. There's two types, HABs and NABs. HABs, as you can guess, are harmful algal blooms. NABs are the kind of the toxin-producing types, the nuisance algal blooms. I'll talk a little bit about what are the algal toxins that we're concerned about and what are the algae from which they derive. And then I'll spend a fair amount of talking about why are they a problem. And here's where we get into some of the One Health relationships, as I mentioned in this tripartite structure. So I'll address each of those as we go along. And as I like to do, I really enjoy translating the science that I do into some kind of practical solutions oriented. It could be policy, it could be best management practices. I play a lot in agricultural landscapes, so I'm very interested in BAPs, and I'll have I'll talk about this a little bit. So I'm going to talk about how we can manage the problem. Okay, I want to start with this comment here, because this is really fundamental to understanding, believe it or not, the connection to harmful algal blooms. That is, by the year 2050, it is projected by the United Nations that we are going to be in and around 10 billion people on this earth, and that we're going to have to produce food for those 10 billion people. In fact, our food production is probably going to have to go up in the order of about 50% just to meet those demands. Notwithstanding the food supply issues that we're having right now and so on and so forth, these are socioeconomic problems that will have to all be factored into this. But this is kind of the backdrop that I'd like you to keep in mind as we're going through here, because part of that increased production is going to be, food production is going to be trying to maximize in ever decreasing space because of urbanization and so on, the land in which we grow crops. We can do that by, for example, increasing fertilizers. That will become more evident as we go through here, the role that they play in harmful algal blooms. So agriculture, as you can appreciate, has a very significant, in some estimates, the most significant footprint from an environmental standpoint. Water use and availability. Somewhere in the order of 70 to 80% of all water is used in agriculture. Greenhouse gas emissions. Habitat loss, all of the things that are particularly important. I'm not going to talk about those today. What I will talk about is the use of synthetic chemicals, in particular fertilizers, because it is the migration, the transport of those fertilizers, particularly nitrogen and phosphorus, from the landscape to aquatic systems that serves as the basis for the production of harmful algal blooms and eutrophication. So I'm going to focus on that today. But again, I just wanted to set the stage that it's really an agricultural problem for the most part. That wasn't always true historically, and in my consideration of the brief history, I'll explain why that is the case. So just again, a little bit of context here. The most significant contributing factor to HABs, synthetic fertilizers, in particular phosphorus and to some extent nitrogen. In freshwater systems, it's phosphorus. In marine systems, it's nitrogen. So where you have your HABs depends on the type, or what nutrient is important depends on what system that you're in. I'm not going to talk much about marine systems today. I'm going to predominantly talk about freshwater systems. So more than half of all synthetic, let's see if I can get the thing working here. Yeah, see, it goes right through. So more than half the synthetic nitrogen fertilizer ever produced has been in the last 20 years. So this is commensurate with this notion that we have to ramp up our food production in order to meet population demands globally, not just within North America and so on. So this is particularly important just as a kind of an indicator. This is the bottom bullet is related to nitrogen, but the synthetic nitrogen has increased by 80% since just 1960. That CO2 statistic, this is about 7 years old, so that's probably much higher, not much, but somewhat higher than 25% now, because every year that increases. Okay, so let's talk a little bit from a historical context, because one of the things I want to impress upon you is that HABs is not a new phenomenon. It may seem like a new phenomenon. The media has certainly picked it up, in particular in relation to places like Lake Erie, where it can be very severe. But it's not a new phenomenon. So the picture on your left here is taken in the 1960s, and this is Cladophora. This is a non-toxin producing species of algae. It's being piled up. You can see it kind of be pushed up into the shores. And in the 1950s and 1960s, Lake Erie was, in many respects, declared to be a dead lake because of the amount of nutrients, particularly phosphorus, that was in that lake. It got so bad because, of course, algae is produced during the summer. Primary cottage season is during the summer. These things would decay and decompose. The stench was so bad that cottagers were selling their cottages for next to nothing because they couldn't stay there, and all they could hope is that they could get a little bit of money for a thing that they weren't using, and or just to stick it out and hope that 5 years, 10 years down the road, governments would find a solution to the problem, which they did, which was good news. But there were a lot of protests at the time to kind of save our lake and clean up Lake Erie. But HABs and NABs are not new, and this is a paper that I discovered while I was putting this talk together. It says, you know, we present a historical account indicating that the advent of intensive farming in the 17th century provided nutrients for promoting harmful algal blooms. So this goes back a long way, recognition of the role that nutrients play in the formation of algal blooms in aquatic systems. In fact, I even found this one, a paper from 1671, which talks about a relation of an inland sea near Danzig, which is modern-day Danzig in Poland, yielding a certain season of the year a green substance which causes certain death. You can see the kind of the wording there. As it was not uncommon at the time, it was published as a letter, not in scientific journals. We didn't really have scientific journals. And they often tacked on other things. So white amber, which is particularly common in the Baltic as a form of amber, was also talked about in this paper. We've known about algal blooms, whether they're toxin-producing or non-toxin producing types, for quite some time. So I wanted to, you know, the teacher in me never really quits, and I wanted to kind of put this in here just to kind of help explain why nutrients running off the landscape can contribute to excessive algal blooms, whether they're toxic or not. And Justus von Liebig in 1840 basically developed what is called the Liebig's Law of the Minimum, which says that your plant growth, translate your crop production, will only be as strong or as good as the limiting nutrient. In aquatic and in landscape areas, matrices, phosphorus is the single most limiting nutrient. Phosphorus, because it is derived from weathering processes of geological substrates. Unlike nitrogen, which is atmospheric, 70% of our atmosphere, lots of nitrogen to be had, but phosphorus is not that available. So it is a limiting nutrient. He compared this to the staves in a barrel, which the lowest stave dictates the level of the water. So too does the lowest concentration of an element dictate the growth. And that if you increase that element, growth will increase up to the next limiting nutrient or element that's critical in that growth process. Now, surprisingly, he's considered to be the father of the fertilizer industry. So with that in mind, you can kind of get a sense of why phosphorus plays such an important role. And if we go back to some very early landscape clearing, I've used Frains Lake in Michigan because that's in the Great Lakes Basin where we do see a lot of problems. Here's some what we call frontier clearing, some of the very early clearing. You see this incredible spike in the amount of sediments going off. And we know this from sediment cores. We can core into that lake and we can look at the strata of those cores. And we can see the amount of deposition linked to any particular year, which we can estimate largely through isotopic analysis. So there's a couple of clearing events that led to this. But what's really important here is that that level compared to when everything was, you know, there was lots of plants on the landscape, lots of trees, they served to take up those nutrients that were in the soils and recycle them back to the nutrients. Nothing or very little was being transported to aquatic systems. Of course, there'd be some. But now we've got a much elevated level, and we often refer to this as the agricultural effect. So this is kind of the modern day context of where we are today in terms of just through sediments and that sort of thing. We augment that because, of course, we add synthetic fertilizers to crops. So in the 1950s, coming back to Lake Erie, as the upper here, this upper level, again, my pointer doesn't work, but this is Put-in Bay, which is in Ohio. A little hard to tell because it's black and white, but that's kind of a mat of algae all in there. So it's an indication of just how significant. These weren't necessarily toxin-producing types, although they did exist at that time. Mostly, as I say, it was the Cadophora form, which is a non-toxin producing type. And the media often declared Lake Erie to be a dead lake. Today, the media does the same thing, but for very different reasons, which we'll talk about in a minute. So at that time, it was a dead lake. And partly because this important indicator organism, which you may or may never have seen before, this is Hexagenia. It's a burrowing mayfly. It is so prolific that weather Doppler radar systems, when they emerge, they are of an order called Ephemeroptera. It means they're very brief. They live for 1 or 2 days. And they emerge. They are picked up on Doppler radar. They almost look like weather systems coming through. They're so prolific. It's amazing to see. I was going to put in some things, but I've only got so much time to show these sorts of things. This was a critical food base for fish in the Lake Erie system. And so when they disappeared, the fish started disappearing. The fact that there were significant anoxic events because of decomposing algae also caused the loss of fish. So yes, there was a tremendous loss of biodiversity in that lake system, and you can understand why the media would refer to it as a dead system. But in fact, it wasn't entirely dead, but of course there was tons of algae, and algae are alive. So it was a very sick system to be sure. And the reason for this, if you look at the catchment area of Lake Erie, is that it is predominantly agricultural, and it has been for almost a century. So when you've got that kind of level of agricultural intensity and all the associated application of fertilizers and loss of sediments and so on, you've got tremendous amounts of nutrients being poured into that system. Factor in the fact that there's about 12 million people in that same basin. And in the 1950s and 1960s, the waste treatment facilities at that time were largely primary treatment, just settling. There was no biological treatment which would help to remove those nutrients. You had phosphates and detergents which were going through those systems. The combination of all of these things just meant that there were tremendous amounts of phosphorus, and the algae grew. And there was some of the problems that we experienced, as I indicated before. So this is a field, for example, where we're applying manure. We do the same for human waste in terms of biosolids. We apply synthetic chemicals. We have best management practices, and I'll talk a little bit this more toward the end of the talk, but the problem is we kind of circumvent them a little bit. So for example, we now have about 50% of our province which is tile drained, almost a direct conduit of those nutrients moving down in the soil profile and then out into a local ditch and ultimately into a stream. Or we have very weak buffering systems, which when you have runoff carries all that sediment nutrients into streams. So while we have best management practices in place, they don't always work. More about that a little bit later on. And the result is what you see here. These are kind of we call it non-point runoff stressors for P, phosphorus and nitrogen on either side. The hot spots exist in the lower Great Lakes, Ontario, and in particular Lake Erie. So this is terms of the contributing tributaries into the lake. Ontario looks bad, and yet we don't see the same degree of algal blooms in Ontario. Why? Because it has a much greater volume than does Lake Erie. It's much, much deeper. It might be a little bit smaller in size, but it has much more assimilative capacity, essentially. And I don't have too many kind of semi-complicated data slides, but this is potentially one of them. But what I wanted to show here is this kind of U-shaped curve where this was the problem that we were trying to address in the 1970s by taking detergents out of water, or out of phosphates out of detergent, by upgrading our sewage treatment plants, on which both efforts we spent billions of dollars, and improving in our BMPs in the manner in which we practiced agriculture on the landscape. And the result of that was to drive down the amount of phosphorus that was being put into these systems. It's the dissolved form of phosphorus, not the bound form, that's really important for uptake by plants. But more recently, we're starting to see an uptake in that activity. We're starting to see an uptake in the amount of dissolved reactive phosphorus. And there's all kinds of different reasons for this. Climate change, I'll explain in a moment, does play a certain role. The actual formulation of the fertilizers, you can go back to the Oklahoma City bombing in 1985. And prior to that, fertilizers were produced using liquid ammonia or liquid nitrogen forms in terms of creating urea. But because of the easy access to that, they then changed that to a granular formulation, which is more susceptible to running off and delivering those components, nitrogen, phosphorus, and other elements alike. And so we're starting to see a significant increase in the amount. These are 4 tributaries coming in out of Ohio. The Maumee River is particularly important in Lake Erie, accounting for about 70% of the phosphorus driving oil blooms in the western basin of Lake Erie. So I have a real problem. I'll talk a little bit about some of the issues we're facing with respect to best management practices in a moment, which are also contributing to that. And so we see a lot of this. Over the last decade, almost every summer, NOAA takes satellite pictures somewhat like this that show, at certain times, algal blooms. And you can access all of these pictures online if you go to the NOAA website. And there's been a lot of reports. The most recent State of the Great Lakes report talks a lot about the HABs problem, as it does all the problems facing the Great Lakes. The International Joint Commission, which is a really good example of kind of transboundary policy development and cooperation, which you don't see a lot of these days. It's a really good kind of governance mechanism to make sure that because in the Great Lakes there's over, if you consider municipal, state, and federal on both sides of the border legislation, there's over 1,000 different pieces of legislation governing aspects of the Great Lakes water quality. And so some of them conflict, some of them don't, some of them at different levels, and so on. So there's a real disparity. So the International Joint Commission really tries to kind of smooth that out and try to make more consistent some of the policies that exist there. Okay. That's a little bit of history, a little bit of background. Now let's talk a little bit about the toxin structure and classification. You can see from here, this is microcystin LR. It has a relatively complicated or large structure. And in fact, there are several classes, which the microcystins, and in particular microcystin LR, is the most common. So in the Great Lakes, for example, microcystin LR probably is in the order of 60 to 70% of the toxin form that we see. But you can measure nodularins, cylindrospermopsin, saxitoxins, anatoxins, lyngbiatoxins. That's more of a benthic one. I'm not going to talk about benthic algae today, but they're becoming recognized as more as pretty important with respect to toxins, and lyngbiatoxin may be one of those, at least in the Great Lakes. And anatoxins as well in some of those benthic algae. In fact, there are about at least 250 different congeners, as we say, different forms of toxin variants. Some of them would be in marine systems, some of them in freshwater systems, some are common to both. There's a lot of different toxins that we're dealing with here. And these are some of the main organisms that you'll find in a place like Lake Erie or the Great Lakes, in which and the types of algae that produce these different types of toxins. So for example, won't go through all of this, but Microcystis, for example, produces both microcystins and anatoxins. Microcystis aeruginosa, which is the most common form or type of microcystis, or the microcystin producing organism, is about 70%. But it produces anatoxins as well. And the relative potency of those, and I'm not going to get into a lot of discussion about the relative potency, somewhat contextual difference, somewhat based on the species that's being exposed to it. Some show more or less sensitivity. But for the most part, anatoxins can be a little bit more toxic than microcystins. It's just the microcystins are much more prolific. So you can kind of get a sense. And algal taxonomy is not particularly easy. I mean, you've got your cellular forms, you've got your filamentous forms, but these all kind of look similar. So trying to get under a microscope and seeing what they actually are is challenging. I haven't shown here, but there's also colonial forms. And they're all adapted. I mean, cyanobacteria are one of the oldest organisms on Earth. They've been around for 2, maybe 3 billion years, and so they're highly adapted to and occur in a wide variety of different environments. And they've been able to evolve under different conditions. For example, anatoxins and cylindrospermopsin producers tend to prefer cool temperatures. In the context of global warming and the fact that our region, Great Lakes Basin, is projected to continue to warm, probably favors those species like microcystins that prefer kind of higher temperatures. And so what we're seeing is microcystins, like Microcystis aeruginosa, are becoming, not microcystins, but microcystis, are becoming increasingly prevalent as in terms of their contribution to these toxin-producing classes of organisms. So it almost really doesn't matter what your conditions are. I mean, we could be in a global cooling phase, and it just means that the microcystis forms would disappear, but maybe now these forms would start to increase. So we're always going to have an issue potentially with respect to these organisms if we keep dumping nutrients in that serve as the basis for their growth. So again, kind of a large table. What I really want to talk about here is just briefly mode of action. These things affect acetylcholinesterase, so they're an enzyme inhibitor, some of them. Some of them prohibit protein synthesis. Some of them inhibit protein phosphates. That's the primary mode of action for microcystins. So I just wanted to kind of give you a sense of what the mode of action for these compounds are. Marine systems slightly different and so on, but some of them are the same, but some of them are also unique for those systems as well. So if you're interested in that, you can kind of go there. And there's a lot of different ways to detect them. I'm not going to talk about today, but we developed one based on a Lemieux oxidation method, which is far more sensitive than the ELISA. So a lot of the historical papers are based on detection through ELISA, but it's relatively insensitive. And a lot of governments are still monitoring using ELISA, and we're trying to get them to monitor using these more sensitive approaches. And in terms of symptoms, whether you're a human or a pet or a livestock here, there's different ways in which can affect. And basically divided between is it external exposure or is it internal exposure? So if you're swimming in a lake or a pond or a stream, but you're not ingesting any of the water, you're not presumably eating any of the algae, then you're going to have external exposure. So you're going to get things like, body. So you're going to get blistering, skin rash, burning, fever, muscle aches. You may or may not experience any of this, but if the exposure is sufficient, because in toxicology, dose makes the poison, there are thresholds below which you might not see an effect, you might experience some of these sorts of symptoms. If it's unfortunate enough that you are to ingest it in some way, and I'll show an example through blood infusion how that actually did happen in what is the most serious and kind of well-known case of toxin poisoning, you are going to experience things like abnormal kidney function or especially liver damage, because these are first and foremost hepatotoxins. They're also neurotoxins, some of them, and so you can get this kind of things like respiratory paralysis, or you can get kind of spasms that you might if you're seriously affected. And we don't know that as much from humans because we haven't seen a lot of situations where humans have been exposed internally, but we do see this a lot in pets that go and frolic and play in a local stream or lake. They drink some of the water, and within minutes to an hour, they've gone through this kind of succession of they lose balance, they become spastic, and then they go through this respiratory paralysis and die. And there are lots of examples. We'll talk about some of those in a moment. So with that, I'll move on then to what are some of the risks to, I've talked about some of the symptoms, what are some of the risks to humans and livestock, and I'll talk a little bit about this together in the same area. This is taken from Lake Taihu in China, where they have serious problems with both toxic and non-toxic. I don't know if this child is swimming in a non-toxic or a toxic one, but it looks like he's got a mouthful, which is not a good idea. So I alluded to this a little bit earlier in 1996 in Brazil in Caruaru at a hemodialysis clinic. 100 people died and 52 people had confirmed exposure to the hepatotoxic microcystins. They did do some follow-up analyses and they did detect the toxin, so they're pretty sure they have kind of that cause-effect relationship established. But they do know that they were being treated with water that was ineffectively treated from a lake known to harbor cyanobacterial blooms. So this is probably the most famous example of what we believe to be algal toxin-induced toxicity in humans. we do see, coming back to Lake Erie, a number of situations where there's lots of potential exposure, and this is kind of underscores the hazard and risks associated with people living in this area. People like, Toledo, Cleveland, Akron, Ohio, even Windsor, Ontario, and Detroit, where you might get exposure. And one of the most famous examples happened in Toledo in 2014, whose water intake is somewhere in right about here. So you can see it's right smack dab in the middle of the algal bloom, which did contain toxin-producing forms. And what it led to is, a 3-day ban on water usage for 500,000 residents. That's a significant inconvenience, to say the least. And they were lined up, and they had to bring in the National Guard in order to keep order, because people tend to go a little squirrely when they're told they can't use their taps for drinking purposes. Turns out you can for bathing purposes if the toxin levels are particularly low. But the problem is, at the time, they didn't know. They had to do the analysis. They did, and back then they were still using a lot of ELISA, which isn't particularly sensitive. So there was a lot of uncertainty. They just said blanket ban. Let's not use the water for any purposes until we give you the all clear. And so we're going to see more of this type of incident in the Great Lakes region, and in particular the western basin of Lake Erie. And you're starting to see more and more on municipal websites, for example, some of the warning systems. So in the middle, for example, on the right-hand side, kind of a red, yellow, green light system where you can go and check. You can use this not just for cyanotoxin, you can use it for bacterial contamination. Beaches use it sometimes as well. But just warning systems that people can go and say, well, what is the situation right now? I know we're in the middle of a harmful algal bloom here in the Western Basin, so let's go to the website and check that out. And then you've got kind of kind of an emergency survival guide. And the one thing that we often think about when we've got microbial contamination, and you could argue that these are kind of microbes of a sort, is to boil water. We have boil water advisories all over Canada and North America. The thing about this is it actually lyses the cells, releases the toxin, and the boiling does not break down the toxin. And so you're actually making the problem worse. So here's one situation where a boil water advisory isn't going to help you. So that's not the recommendation if you're dealing with harmful algal blooms. So I mentioned before that there was all kinds of examples. I spent like 10 minutes online and came up with some of these examples where dogs, people talk about, and very distraught people, because of course pets are so important in our lives, but they, and the sense of helplessness, because within an hour the dog was dead and there's nothing they could do. Some of them couldn't even get them to the vet in time. So it's very, very rapid onset of symptoms and mortality. But it's also the case with livestock as well. And one of the examples here, who knew there was a journal or a clinical studies journal called Flock and Herd, but there is. And they were talking, they're describing an incident where you can see all these kind of sheep here. Now just an hour or two or so before this, those sheep were alive. So they had probably been grazing and migrated down to this pond. A little hard to tell, but there's a green hue there. It had just been treated with copper sulfate, which is often used to treat algae. But the copper sulfate would potentially release the toxins by lysing the cells as they died. So you're actually creating a greater problem if you've got, eventually the toxins will disappear and they'll break down or they'll become entrained in the sediments. But the timing in this case was everything. So you had these dead animals that had drank from this thing. There's also some suggestion that perhaps even the copper sulfate itself, which is known to have a certain degree of toxicity depending on how much you apply, could also relate to or cause mortality. All right. So that's a little bit about the risk to ecosystems and animals. I think you could probably do a talk on any one of these angles here. So I'm just kind of giving you a flavor for that. I tend to play a little bit more in the risk to ecosystems. So here I'll get a little bit more detailed in some of the considerations that I have. I won't spend a lot of time here. You can find these sorts of diagrams. The main thing here is to notice that the numbers here represent the concentrations of toxins that were measured in these various ecosystem components. Number two, that the arrows indicate that there's toxicity toward each or a lot of these components. the benthic organisms, the fish, different types of fish, filter feeding mussels potentially accumulating the toxins if those are consumed by other organisms, like a raccoon, for example, and even to invertebrates where I've done some work. So the point is that there's a lot of potential effects at the ecosystem level, a lot of particular components. And in a place like Lake Erie, where you have these broad and widespread events, the question is, what is it doing to those ecosystems? And so those are some of the questions that we kind of like to address. So one component of the food chain of the Great Lakes are cladocerans, or water fleas. And from a toxicity standpoint, we use this particular species called Daphnia magna. And what you see here will make sense in a moment. These are just different average sizes of 3 different treatments: the control, the microcystin, and the non-toxic microcystin that you see there. And this is some of the data that we generate. And the hypothesis we were trying to test here is, we know that there's toxicity because some previous work showed that we had toxicity that we observed in this organism under single generation exposures. The question is, what happens over multiple generation exposures? These guys can have 3, 4, 5, half a dozen generations a year. So what happens when they're in an algal bloom? Some algal blooms only last a couple of weeks, some a month, 2 months. So it's theoretically possible they could reproduce several times during that period if they're not wiped out by the bloom. And what we found in this graph here is that, in fact, this is the control level here. So this is the controls, a little bit of variation, but pretty consistent. We did, in fact, find up to the 4th generation, at least, that there was a tendency to kind of adapt, if you will, to these toxins. But then all of a sudden we saw in the 5th generation a significant drop. And if you kind of look at the size of that, if you assume that there's a relationship between the size and sensitivity or reproductive output of those organisms, we don't really see a significant, in fact, it's a lot larger than the 4th generation one, which produced a lot more. So there's other things going on here that we're not so sure about. And even in the mixed or in the non-toxic, we saw the same sort of phenomenon, just the middle generation produced a whole lot more, but it wasn't necessarily well correlated with size. So we need to explore a little bit further what exactly is driving this, but it is somewhat comforting to know that there may be some evidence of maternity transferable toxin intolerance. And in fact, given that algal blooms, toxic algae, have been around for millions, billions of years, and these systems have evolved in the face of these toxins, it does make some sense that the organisms exposed to them would also evolve mechanisms to deal with it. But the pace of change, the rapidity with which the toxins are being produced now over very short periods of time, are probably not something that they've evolved mechanisms to deal with. So that's a bit of a problem. And one of the things that we're starting to look at is, the gut microbiome. So, for example, in this study, this wasn't our work, but there was evidence of a high occurrence of what is referred to as the MLRA gene. So that's a microcystin degrading gene in a copepod microbiome. They were exposed to nodularin-producing cyanobacteria, but was thought to be an evolutionary adaptation to be able to withstand the effects of cyanobacteria, and that those organisms that were capable of doing that and passing on their genes through reproduction would in fact be able to pass those genes on, so confer increasing resistance over time. We don't know if those mechanisms can happen, again, with the rapidity with which toxins are being produced in the Great Lakes right now with these algal blooms, but maybe not. We'll have to see. These are open questions. One of the approaches in the toxicology world that people like to use now is adverse outcomes pathways. So for the longest time, I grew up as a population community ecologist, and I always knew it was important to understand what the mechanisms were and what's happening at the suborganism level thing. But there was a period of there was a period of time where people started just measuring biomarkers, changes in enzyme levels, changes in, and now we've got the omics, the various omics, genomics, proteomics, metabolomics, so they're measuring levels of everything. The big question is, okay, you've measured change, so what? That's an indication that there's been an exposure, but does it correlate in some way to an apical endpoint like growth or reproduction? So the adverse outcome pathways approach allows you to do studies that take start at the molecular initiating event and work all the way through to the adverse outcome. And this is one of the things that we tried to do with respect to the organisms that we're looking for. Not so much with the invertebrates, but certainly with rainbow trout and lake trout as well. And I can't, I mean, I could show slide after slide of data that we generated, but what I'll do is just show an example of the shotgun proteomics outcome. And yeah, there's a lot of information here. I mean, you've got the gray or the statistical p-values. These are the fold changes to a low and high exposure in muscle and liver to rainbow trout and lake trout. I'd probably take about 10 minutes to parse out all the details of this graph. But what I really want to point out is that a lot of these, shall we say, upregulated genes relate to things like cancer, apoptosis, which is programmed cell death, reactive oxygen species, cell damage, and DNA repair, amongst other things. So there's an indication that these exposure to these toxins are, at least at the molecular level, causing these changes. And then the question is, does that affect growth? So we've looked at histology, we looked at growth, we've seen effects, which we've been able to trace back at least to these changes in the genes, not necessarily specific ones, but we have, for example, seen evidence of cancer in some of these fish and so on, which may or may not have. We haven't taken it far enough to really know and say, yes, there's an absolute cause and effect here, but we are at least comfortable with knowing that there is some connection here. And of course, what it means is that there is uptake by the organisms into different organisms. And again, it's just easier to cut and paste a whole table and then kind of direct you to the important parts of what I want to see here. So basically, these are the concentrations that we measured in liver, kidney, and muscle. And just see, you look down this column and this column, they're relatively high compared to the muscle. And one of the questions that we were really interested in asking was, are there implications for the consumption of fish? We don't typically eat the offal or trimmings, that is the liver or the kidney, but we do eat the muscle. So this suggests that there's probably, and you know, we're in the process of helping the Ontario Ministry of Conservation, Environment, Conservation and Parks. They've changed their name 3 times since I started here at the University of Guelph. As to whether or not they actually need fish consumption advisories. So that's kind of a human health context. And so that was in a lab study, but if you go out into the lake and you start measuring in different organisms, which we did in conjunction with said agency, you can see that in terms of total microcystins in the liver, quite high, with some significant ranges here. Look at the level there, but relatively low in the muscle. That's good news from a consumption of fish standpoint. So we conclude that the risk via consumption of fish muscle seem to be relatively low. And that's really important for perch. I was down on Lake Erie. There's in Port Dover, there's Callahan's Beach House, and I had the perch dinner there the other night. I'm not too concerned about toxins being, in exposure. I'm still here, and I don't have any twitches or anything like that. Okay, so we're in the process of publishing this. This is a preprint. If you're interested, you can kind of go and read the paper. Kind of low human risk of algal toxins from consuming fish. In this case, in Lake St. Clair, but we've also looked at Lake Erie. All right, so the World Health Organization has set certain water quality criteria. For drinking water, it's 1 microgram per liter. For exposure recreationally speaking, a local stream or lake, it's 10. And provisionally, in terms of a tolerable daily intake, the value is 0.04 micrograms per body weight. So our data suggests that that's probably pretty reasonable estimate, and it looks like our colleague at the OMECP is aiming at, I've heard 0.06 as a possibility there as well, so we'll see what comes out of that. Our question was, do we even need them? The question that I think we concluded that, well, we're going to develop them, but like everything, I mean, we have fish consumption advisories for mercury, for PCBs. You just have to be safe about it. You can still eat those fish. You just don't want to eat the older fish, or don't want to eat a lot of fish. That poses a bit of a problem for indigenous communities where fish might comprise a very significant part of their diet, but you just have to kind of be careful. Okay, so I'm going to finish off talking about how we manage the problem. And again, I'll start with this comment here. It's not really hyperbolic, even if it reads a little bit as such. We are at a make or break point, or a moment for the planet. This from the chair of the Commission on Sustainable Agriculture Intensification at the United Nations. We need to produce more nutrition and healthy food, while at the same time reversing the environmental degradation. The very problems that we have that are leading to this problem of harmful algal blooms in aquatic systems. So they talk about other things like malnutrition and poverty. Of course, these are things that we need to address as United Nations, so they have their 17 sustainability goals and incorporate them all into, or a lot of them into statements like this. So how do we go about doing that? Well, I mentioned before BMPs and the implementation of those in an agricultural context. We talked about the elimination of phosphates and detergents. That's one of the things we were able to do to counter the problem back then in the 50s and 60s. We have at our disposal a number of tools, and this is slowly evolving. I have to say that the farming community is very reluctant to engage in regulations. They hate regulations. So there's a few of them under the Nutrient Management Act. I mean, now you have to justify the application of fertilizers by measuring the amount of nitrogen, phosphorus in the soil. And if it's above a certain level, which is known to support plant growth, then you really don't get to apply those lands. So you have to be targeted. And there's all kinds of documents produced by the Ontario Ministry of Agriculture, Food and Rural Affairs, which talk about best management practices. This is, info sheet number 17. There's dozens of these things that you can avail yourself of. One of the more recent things that you're seeing more and more in the literature is the so-called 4 R's, principles of nutrient stewardship. So the right source, the right rate, the right time, and the right place. So for example, matches amount of fertilizer to crop needs. Well, this goes right into measuring what's in the soil so that a farmer knows, okay, things are pretty good this year, maybe I don't need that. Or that amount, or I can reduce that amount. Now, you may be familiar with the federal government's recent policy to say that we're going to try and promote a 30% reduction in nitrogen emissions from agricultural areas. In the Netherlands, what they did is they said, we're going to do a 30% reduction in fertilizer inputs. But in Canada, we're saying we're doing a 30% reduction in the management of fertilizers through the use of these sorts of approaches, which will lead to a 30%. So it's a fundamentally different approach, and it's, I think, the right approach. Despite what some on the farming community on the right political side of the spectrum will tell you, it's not the case. We're not reducing. But the neat thing is if you do these sorts of things, you follow these sorts of practices, you may get a 20 or 30% reduction in the use of fertilizers. But it's not because you had to, it's because you're just changing the way in which you practice. And of course, it's the kind of the usual suspects. We have no-till, reduced-till, crop rotation, cover crops, all things that keep organic matter on the land. That's good for building up organic matter in the soil, which helps to process those nutrients. But it also keeps erosion from removing some of the sediments and associated nutrients from the soil. One of the things that's very, very common, or commonly touted at least, but not very common in agricultural landscapes, is this use of riparian buffers. The idea being that waters eroding off the landscape will be taken up by those trees or whatever vegetation happens to be there. The problem is a lot of agricultural landscapes don't look anything like that. These fields go right down to the edge, or maybe there's just a little small 2 or 3 meter buffer there. So it's not particularly effective. And now, as I indicated before, we have 50% roughly of the Ontario agricultural landscape is tile drained. Those tile drains completely circumvent the utility of those buffers for the most part. So buffers aren't really the way to go as much anymore, although in terms of habitat and that sort of, there's still a lot of merit in doing that. Our governments don't really provide the incentives to farmers to do that, so the farmers say, well, why should I? I mean, I'm losing crop space. I'm not being compensated for that loss. Well, I'm not going to plant stuff that takes away from my rotations. So we are, despite all that, still seeing these kind of algal blooms in Lake Erie. Well, I've talked about some of the factors that led to that before, but there's some other issues too. I just wanted to throw this in here. This is, and I realize this ends in 2011, but my understanding is that trend hasn't changed at all. So the original objective for Lake Erie was to limit phosphorus inputs to 11,000 metric tons. That really wasn't making much of a difference. So what they said was, we're going to now change that, and we're going to aim for about 6,000 metric tons, a 40% reduction in phosphorus inputs. Well, you can see that for the most part, we don't really hit that, and often we exceed it, and that seems to be continuing on. So whatever we are doing, we're not doing it enough. Part of the problem is that in Ontario, only 40% of farms have an environmental farm plan. This is a volunteer thing. So a lot of, less than half of farmers actually do it, which has certain things that you need to practice or do. We, as I mentioned, have tile drains, so that's circumventing some of the problems. You can see the kind of the edge of field thing here, very little riparian area to take off anything that's coming off the field. This is this is an example of a bad best management practice. The BMP for spreading manure says do not apply on frozen or snowed soils. But there it is happening right there. And what happens when that snow melts, it can carry a lot of that material into the aquatic system, especially if it's a rapid melt or followed by, the next day a very solid rain or something that kind of carries that stuff off. So we have subscription, we have application of BMPs, but we don't necessarily have enough of it in order to make the kind of difference that we need to get down to that 6,000 metric ton objective in the Great Lakes. So one of the papers that I wrote with my PhD student Renee was what we call kind of the sustainable agricultural imperative. Yes, we talked about some of the BMPs and why they're failing and that sort of thing, but what we really wanted to talk about was this whole idea and kind of a new area, if you will, in terms of what we call sustainable intensification of agriculture, incorporating practices which range from regenerative agriculture, I'll talk about that in a moment, to things like integrated pest management when you're dealing with the control of pests and the introduction of pesticides onto the landscape, where you have these you can look at the variation there. Whenever you've got this degree or type of heterogeneity on the landscape, you're improving habitat templates, which means you can attract more diversity to the landscape, which builds ecological resilience into that landscape, which means that you have greater capacity to take up a lot of those nutrients in the case of the causative agents of harmful algal blooms. So this whole notion of sustainable agriculture is something that in principle farmers say, yeah, it's great, but what's it going to cost me, or how do I do it, and that sort of thing. And part of that is this idea of regenerative agriculture, kind of an integrative system of farming principles and practices, which do all kinds of good things. But basically what the most important ones are building back soil organic carbon. There are those, and I'm not in disagreement with them, that suggest that the loss of soil health may be the single most important factor contributing to problems with feeding the world and food security in modern times. So part of the loss of soil health is the loss of organic material. So if you can do any kind of practice that maximizes the amount of organic matter you can build back into the soil, and the associated microbial community that can then process in association with that the nutrients and so on, this is kind of a good thing. And it incorporates some of the best management practices that we saw before, like cover crops and crop rotations and so on. Diversity of crops, these are all good things. Seeding the soil with arbuscular mycorrhizal fungi to enhance those soil root plant relationships so that you have greater capacity for incorporating nitrogen and increasing your crop production. That's not something a lot of farmers do, but I've talked with farmers who do practice that, and their results are quite astounding relative to, say, some of their neighbors. But mostly on a small scale, so a lot of the problem here is scaling up to the size, the industrial scale that we need to. That's the big question right now in terms of applying these kind of core principles to kind of understand context, minimize soil disturbance, maximize crop diversity, this is crop rotations, keep the soil covered, so there's cover crops, maintain living root year-round, livestock, integrate livestock. So, have your livestock grazing on those systems, returning those nutrients in the form of their fecal material back into the landscape, not just putting it in the corner of your barn and then, spreading it off through some high carbon output, tractor-based mechanism. Let the cattle do that themselves. And it looks a lot in many respects when you talk about, economic and social resiliency, the soil health, the water health, the increased biodiversity that comes with this in practice, again, mostly on a small scale at this point, kind of cow and herd well-being. Looks a lot like a One Health approach, really, to kind of solving this problem. You look at that and think, well, yeah, okay, well, what's the point? What's the look and to harmful algal blooms? Well, the point is that if you do that, you don't have the nutrients moving off the landscape in order to create the harmful algal blooms that are in the systems that we have or see on an annual basis. So this is really hard to see. This is the kind of the diagram that we produced for the publication that I showed a little bit earlier. And basically the goal is to create these resilient ecosystems, and you have to focus on not just the environment. If I'm interested in harmful algal blooms, yes, most of my work is in the form of the environment. But in the true context of the United Nations sustainability goals, you want to focus on the technology that allows you to do these things, the culture, the economics, the diets, the policy. All of these things have to become part of this integrative system, and that's what in regenerative agriculture kind of allows you to do. And that kind of integrated approach really does sound a lot like One Health, as we say. Okay, questions?